So thank you all for joining us again. We enjoyed a wonderful presentation by Julie and Christy from Leahy Medical Center. So we are thrilled to now have our next presenter. And I'm very excited to introduce our next presenter, Dr. Sabrina Paganoni. Dr. Sabrina Paganoni is an assistant professor at Harvard Medical School. She obtained her MD degree at the University of Milan in Italy and a PhD in neuroscience at Northwestern University in Chicago, Illinois. She completed her medical training at Harvard Medical School in Boston, residency at Spaulding Rehabilitation Hospital, and fellowship at Brigham and Women's Hospital and Massachusetts General Hospital. Her clinical and research activities relate to her subspecialty interests of neuromuscular medicine and clinical neurophysiology, or EMG, with a particular focus on ALS. In addition to her clinical activity, Dr. Paganoni is actively engaged in clinical research. Her research focuses on ALS therapy development. She is the principal investigator of several ALS clinical trials and is interested in using biomarkers to enhance the efficiency of ALS research. She is also passionate about developing innovative assistive technology tools that can help improve the quality of life for people with disabilities. Dr. Paganoni holds 3 board certifications: physical medicine and rehabilitation, neuromuscular medicine, and electrodiagnostic medicine. Dr. Paganoni is a member of the Association of Academic Physiatrists, American Academy of Neurology, and American Association of Neuromuscular and Electrodiagnostic Medicine. Please welcome me in joining Dr. Sabrina Paganoni.
Thank you, and it's great to be here with all of you on this rainy day, at least here in Boston. I see many names, I recognize many names, friends and people I've seen recently and others who have not seen for a while, but great to be here with all of you today. And I think I can share my screen, and I have a presentation here. Hold on a second, let me just start. So today I want to focus a little bit on ALS research and specifically ALS trials, which is now my primary occupation. So I want to give kind of an overview of the field and then focus on 2 trials that I'm directly involved with, the Healy ALS platform trial, as well as the CENTAR or AMX35 trial. And obviously, you know, I want to leave enough time for questions. So as always, please feel free to write questions in the chat, and then hopefully I'll answer some of them during the presentation. If not, we can you know, talk after my presentation, which should be about 25 minutes or so. And I want to start by saying that while 2020 has been a challenging year, obviously for many reasons for everyone, it will also be remembered as a great year of great progress in terms of ALS trials, because we do have a lot of promising trials, you know, opportunities for new drugs to be brought to market. So I think we don't want to lose track of that. And the reason we have so many trials and so many interesting research going on is because of the many years of investment in basic science and clinical science that have identified so many new targets for intervention. What does that mean? Well, the graphic here shows the picture of a nerve cell and a schematic of many different things that can go wrong in the nerve cells. And while there's many things that can go wrong in the cell and then ultimately cause ALS, all of the pictures here represent an opportunity. So an opportunity of intervention, because if we know what's not working, we can develop interventions to target those mechanisms and trying to stop them or even reverse them. In fact, because of this explosion in basic science, ALS is the neuromuscular disease with the largest drug development pipeline. So if you just compare ALS, which is indicated by the arrow to the left, to all the other neuromuscular diseases, muscular dystrophy, for example, you'll see that ALS has many, many more drugs in development because we know more, because now research is finally providing many new targets for intervention. And so different companies and different investigators are developing experimental compounds or investigational products to try to stop or even reverse the disease. There are many, many trials all around the world that are active and enrolling. They actually act on different parts of the, different cellular mechanisms, whether it's immunomodulation, gene therapy, or other treatments for familial ALS, whether they are stem cell based or small molecule based or monoclonal antibodies, different ways of trying to achieve the same goal, which is to treat ALS, to develop as many treatments as possible for both familial ALS as well as sporadic ALS. Now, if I underlined some of these compounds, because those are the ones in phase 3. So if you're familiar with drug development, whenever we develop a new compound, let's say that we have a new idea, a new target for intervention, and we develop a new drug, we need to test it according to a certain kind of schedule. There's a phase 1, phase 2, and phase 3. So the good news is that there's at least 6 or 7 trials right now all around the world, including the United States, where these drugs are now in phase 3. And that's important because when you are in phase 3, that means that you are actually testing the drug in the late stage of development. You are actually testing whether the drug is effective. And if the phase 3 trial is positive, if there are good results, it's actually possible potentially to bring a new drug to market. So that's why it's important to have many different drugs in different stages of development. And in ALS, there are many drugs in so-called late stages of development, which means very close to potentially going to market if the results are positive. Now, because we have so many drugs in development, as I just explained, the Healy Center where I work, actually I'm speaking from there in Boston, Mass, at Massachusetts General Hospital. The Healy Center launched a very new and innovative initiative called the Healy ALS Platform Trial, which is sponsored by the Healy Center at Mass General, but also leverages fantastic collaboration from many different groups, academic groups, foundations, and industry partners, including and foundations, including the ALS Association that is supporting, helping to support the trial. So it's a really phenomenal collaboration that's going on at different centers in the US. And that's the first topic of my talk. I want to explain how we're trying to leverage this trial to really change the pace of ALS research. I told you before that there are many treatments in development. And so for us, the question is, how do we test as many as possible, as quickly as possible? So the Healy ALS platform trial exists for only one reason: to accelerate the development of new drugs for ALS, because we know we have many in development and we want to test many and quickly so that we can, if they're positive, we can bring them to market. If not, we can move on and test more. In fact, normally in traditional ALS drug development, but in general in medicine, normally, traditionally, we test only one treatment at a time. So what happens is that, let's say that a pharmaceutical company develops a promising compound, treatment A, for example, and they decide to go the traditional route. So what they have to do is to build an entire infrastructure to test that one treatment and answer that one scientific question. By way of analogy, it's as if every time we need to play a football game, we had to build a stadium just to play that one game. Instead, with the platform trial, we're doing something dramatically different and really new and innovative, which is to use the same infrastructure, the same stadium, as an open platform to accommodate as many treatments as possible. So what we did was that over the last year or so, we spent time building this infrastructure, this platform that we think is an optimized infrastructure to test as many drugs with ALS as possible. We started with 3 treatments over the summer: A, B, and C, and we are really just a few days away from starting to to test the fourth treatment, treatment D. And I'm going to explain what they are. So I just want to start by acknowledging the contributions of a dedicated group of investigators and statisticians that have contributed in different ways to the establishment of these trials. So this took many months to set up because it's a very large and ambitious project, but now it's up and running, and I think really will change the pace of ALS research. Many different investigators contributed to the development and are now working to run the trial. I just want to acknowledge first and foremost Dr. Mary Sukovic, the director of the Healy Center at for ALS at Mass General Hospital, who's really the leader and the person who had the idea first and the vision to do something so innovative and so important for the field. And so she brought us together with all the investigators and statisticians whose names and pictures are on the slide, and we've been working together as a team to really design this infrastructure that leverages the expertise of many experts in the field. So all the people on the slide contributed their own knowledge, whether it's in biostatistics, genetics, biomarkers, other clinical practice, all sorts of aspects of ALS care and research to make sure that we could design the best possible trial. And now we're able to run this trial thanks to a very large clinical operations team. Because of the size and scope of the project, we have a large team dedicated 100% to this project, because again, it's really something that can change the field and change the pace of drug development for ALS. So, and it is already doing that. So we have a very dedicated team, and I just want to thank everyone on the slide and all our collaborators for really making this trial possible. We've been working with patients from the beginning. We have a patient advisory committee, and we are really grateful for all the support that the patients have given us and all the wonderful ideas. So many features of the design of this trial and many things that we're doing, such as doing weekly webinars with the community, etc. All of these came from patients. So patients asked us to include certain features because they're more patient-centered, and we were absolutely delighted to do so and partner with them. And we were very happy to especially collaborate with IMALS and their group, and ultimately we received a 5-star rating because of the patient-centered design of the trial. We also collaborated with many other stakeholders. We selected 54 sites, meaning ALS clinics all around the country. They are indicated here on the slide, and we are connected by one coordination center, including one central IRB, which is basically the ethics committee that oversees the conduct of the trial. All of these ALS clinics are part of NEALS, which is the ALS consortium. That's a very experienced consortium of sites that's been running studies in the US and elsewhere for many, many years. So great experience from these sites. I also want to mention that we're gonna issue a call for more sites so that we can add more geographies again over the next few months. In order to decide which drugs we're going to test, or we are testing already in the trial, we have a very distinguished therapy evaluation committee that includes the people on this slide who are some of the best experts in ALS science in the world. And they meet regularly to really review the state of the science and what compounds might be the best fit for the trial. So the initial group of drugs that we are testing right now was selected based on a competition. Basically, we issued a competition for drugs that, for companies that would want to be part of this. And we reviewed initially 27 applications. And so the first few were selected from that pool, but now we continue to engage with companies. In fact, we are already working on adding new companies, that just applied so that we can continue to add the best drugs. We've been working closely with the FDA. We went to the FDA a few times, also had virtual meetings with them in 2020 because of the pandemic. And we had very positive meetings. Really, the great support from the FDA for this type of approach. And importantly, we know that this type of trial, because of the specific design that we discussed and agreed upon with the FDA, if the result of the trial is positive, meaning that one of these drugs has a remarkable effect on the disease, these this one trial could serve as the pivotal trial or registration trial. What that means is that this trial could be enough, if obviously the results are positive, to bring a new drug to market. So I think it's really high impact kind of a project, because new drugs could really go to market just based on this trial, if they are effective, obviously. These are the drugs that we are testing right now, regimens A, B, C, and D, and they are developed by different manufacturers. Zeleucoplan, the one in orange, is a drug that targets the complement, which is essentially a part of our immune system. Verdipestat, the second one in green, also targets the immune system, just a different part of it, called microglia, to essentially reduce neuroinflammation in the brain. CNM-Au8 in blue is a drug that works on energy production and mitochondrial function. And then pridopidine, that's the new one, regimen D in gold here. It's the one that acts on sigma-1 receptor, which is an important receptor to basically regulate the relationship between different parts of the cell, the nucleus and the cytoplasm. And so this is very important, again, that we are testing different drugs with different mechanisms of action, because we don't know ultimately which target is the best, but we want to test as many as possible so that we can develop as many drugs as possible. I do want to mention that we are planning a webinar series run by the scientists from each company to explain in great detail all the mechanism of actions and all the mechanisms and all the science behind these drugs. So that's coming up over the next few weeks for people who may be interested. In terms of the study design, this is a placebo-controlled trial, and the placebo-controlled trial lasts 6 months or 24 weeks. Now, one important feature of this trial, which is very different from other trials, from standalone trials, is that because we are using a common infrastructure, that infrastructure I described before, we are able to share placebo among different drugs. So what that means is that we're testing different drugs, and the data from the placebo group are shared or pooled, so that we can reduce as much as possible the number of people who get assigned to placebo. Specifically, the randomization ratio is 3-1. What that means is that for every 4 people that enter the trial, 3 or 75% will go on active drug, and only 1 or 25%, a quarter, will go on placebo, which is much better than standalone trials. I also want to mention that this trial includes the possibility, an optional possibility of open label extension. So if a participant completes the placebo-controlled trial, the participant that then has the option to go on an open label extension. The open label extension means that people who complete the trial can receive active drug for sure for an extended period of time. So while this is a placebo-controlled trial, people that complete the trial are given the option to receive active drug. And we think this is really a feature of a patient-centered design that we really wanted to include in the trial. So far, we have 33 sites that are actively enrolling. Over 200 people signed up to participate in the trial, and we have 143 people randomized within one of the 3 regimens to either active or placebo. These enrollment rates are 3 to 4 times faster than in traditional trials, because normally kind of historically, we know that we generally enroll 1 participant per clinic per month. And in this trial, instead, we have been enrolling at a much faster rate, 3 to 4 participants per site per month. So we're very excited that despite the pandemic, the trial is up and running. And obviously right now, different sites, because of the second surge of the pandemic, are having, are encountering new COVID-related challenges, but we're working with them to really continue the trial, because again, we think it's important for the population, and we all agree, and we're all trying to do as much as possible to really continue the trial. And again, so far it's going very well. Just a couple of words about the design. Every regimen will include 160 participants randomized 3-1 who will be treated in the placebo-controlled trial for 6 months, and then again after that there is an open label extension. These are the criteria for entry into the trial. Everything is publicly available, so please check on our you can take a look at our website. We have all this information on the website, and the website is the website of the Healy Center. We're going to measure different endpoints, both clinical endpoints and biomarkers in the trial. So essentially what we want to do is to really tell if a drug is safe and effective on multiple measures: the functional rating scales, muscle strength, respiratory capacity, and very important biomarkers. As you know, there's a lot of interest in ALS biomarkers, and so we have a number of them, including in the trial, from genetic information to neurofilaments, other biomarkers, voice recordings, and home measures that leverage smartphone apps. So we are really trying to integrate all the most innovative aspects of ALS research into one trial so that we can leverage the trial for two goals. Essentially, number one, obviously the most important goal, is to test many drugs and find effective treatments for ALS. But then there's also another goal, which is to learn more about the disease, because based on these biomarkers, we can really develop innovative ways of measuring ALS. And the impact of that will not only be on these specific drugs that we are testing, but there's going to be a very broad impact on the entire field, because everything that we know will be shared, and it can really help also other projects, not just this specific project. And here I'm reporting our website link. If you're interested in learning which centers are enrolling, there is a complete list. We have several centers in Massachusetts and in New England. So please take a look at the website and you can get all the information. We also have a patient navigator, or actually two patient navigators, Catherine and Allison, whose full-time job is to answer the phone and email and connect with the patient community and have a conversation with the community. If you have any questions about the trial, please contact them at any time. And lastly, we have newsletters. You can sign up for this free newsletter on our website. And we are also hosting weekly webinars. The idea really came from patients. They wanted us to be available to take questions from the community. And actually, in looking at the names on the screen today, I recognize many of you are already joining our weekly webinars. Thank you for your participation, and please continue to come back. They're generally on Thursday afternoon, 5 p.m. Eastern. And we just love to connect with all of you. Please invite everyone you know and post it on social media, because we're happy to answer any questions you might have about this trial or other trials. And again, that's our link. We also do post the recordings and the slide decks. So if you miss the actual live presentation, you can always watch the recording. Lastly, I want to mention that we are working on launching an expanded access program in parallel to the platform trial. So we are working to achieve this goal. The idea came from the patient advisory committee, and the idea is to allow a parallel track for people who are not eligible for the trial. The trial is actually much broader in terms of access and inclusion compared to other trials. However, for statistical and scientific reasons, we cannot take all comers, and we can talk more about it, but we do want to give options to everyone, even to people who are not eligible for the actual trial. And so for that reason, we're building an expanded access program. This is a huge project, also very expensive project. So we are actively fundraising and working to support this effort. So we're hoping to open these in early 2021. Now I want to switch gears and present the results of a trial that has already ended. So the platform trial just started, and it's enrolling really fast, very fast pace. But now I also have other good news, which is the news of the Center study, a trial of AMX35 for people with ALS. We tested the safety, functional benefits, and survival benefits of this drug for people with ALS. And we recently reported the results in September, October. So we're very excited about this. And these are positive results. So we, I think it's very important to communicate these results, because they could really be a great new opportunity for people with ALS. So the AMX story is actually very interesting. The company, Amylyx Pharmaceuticals, was founded in 2013, or a few years ago, by Justin Klee and Josh Cohen, who are phenomenal, absolutely brilliant young men. They were in college when they started the company. They had this phenomenal idea to combine two drugs into one, and that's how AMX35 was born. And then they connected with different experts in the field, both in ALS and Alzheimer's disease. Specifically, they met with Dr. Rudy Tanzi, whose picture is on the slide, who's an expert in ALS, in Alzheimer's disease, and then with Mary Sukovic, who is obviously a phenomenal expert in ALS. And the two of them, helped them basically take the next steps, connected them with Peter Foss from ALS Finding a Cure, and then ultimately to the ALS Association. We wrote the grant together, and in 2016 we received a grant from the ALS Association and ALS Finding a Cure to jumpstart the ALS program. At the same time, Amylyx Pharmaceuticals worked with Dr. Tanzi and others in the Alzheimer's disease area, and they also got a grant to start an Alzheimer's study. So both trials, have been going on for the last few years. The ALS study progressed. Started first and finished first, and so we are very excited to share the results with you today. The Alzheimer's trial is still ongoing, and the results should be available in early 2021. So very exciting developments for both diseases, and really interesting therapeutic that we've been testing together over the last few years. The CENTAUR trial was a phenomenal collaboration in the ALS community. I mentioned the initial grant from the ALS Association and ALS Finding a Cure, with collaboration from many experts from academia, from different universities, different academic medical centers, working together to develop this new treatment and to test this new treatment. All of the centers are also part of NEALS, the Northeast ALS Consortium that I mentioned earlier, and the trial was designed and coordinated through Massachusetts General Hospital and the Barrow Neurological Institute, and it's been a great collaboration between different universities and industry. Amylyx Pharmaceuticals, again, phenomenal industry partner. We've been working closely together for the last few years, and really it's been really the highlight of my work so far, really fantastic collaboration. AMX35, the actual drug, is a combination of two drugs. They're called PB and TERSO. The acronyms are kind of useful, helpful, because the names are very complicated. PB stands for sodium phenylbutyrate, and TERSO stands for terursodiol. So these two molecules, PB and TERSO for short, target different cell components to keep the nerve cells functioning properly. They were already known before Amylyx combined them. So individually, as individual compounds, we already knew that they had neuroprotective properties in lab models of ALS and other neurodegenerative diseases. But what's exciting is that the company tried to combine them, and they saw that when they were combined, they were even better at improving the outcomes and really blocking neurodegeneration. The resulting product, the combination of the two, is called AMX35. This is a product that's taken either by mouth or feeding tube. It's a powder, so it comes in a pouch. Then the person who takes AMX35 will open the pouch, dissolve the content, the powder, in water, and it's taken once a day for the first 3 weeks, and then twice a day. It can have a bitter taste. So in the trial, there was a placebo that was matched for taste and appearance. We reported the results of the CENTAUR trial. CENTAUR stands for the combination of phenylbutyrate and terursodiol. And we reported them in 2 papers that were published, the first in September and the second in October of 2020, so just a few months ago. The first paper was published in the New England Journal of Medicine and reports the effect of AMX35 on physical function. And the second paper was published in the journal Muscle and Nerve and reports the effects of the drug on survival. The trial was made possible thanks to the generous participation of 137 people with ALS who enrolled in the study. Their average age at the time of enrollment was 57, and they were early in their disease course, about 1 year since symptom onset and 6 months since diagnosis. About a quarter of them had bulbar onset, meaning that the first symptoms were in the face, so with swallowing or speaking difficulties. And the vast majority of participants was already on riluzole or edaravone or both when they started the trial. So the results I'm gonna share are results that were generated in a population of people with ALS who was already taking standard of care, where we define standard of care as riluzole and or edaravone. So this means that the results I'm going to show you are on top of standard of care. So we're showing results that are additive to the already known drugs for ALS, riluzole and edaravone. So what did we want to test? Well, we wanted to test the effect of AMX35 on physical function over a 6-month period. We also wanted to test the effects of AMX35 on survival. This was assessed long-term over a 3-year period. And also, of course, we wanted to confirm that AMX35 is safe and tolerable. The two individual drugs, as I said earlier, they were already available before, and we knew that as individual compounds, they were safe, but we wanted to make sure that the combination was also safe and tolerable. The study design, in a way, is similar to the platform trial in terms of duration of the trial. So we had the first randomized period, which was 6 months long, and then we had an open label extension, which was much longer. In fact, it is still going on. We still have people in the open label extension. So during the first randomized period, we randomly assigned participants to either AMX35 or placebo, and we did so in a 2-to-1 ratio. This means that the group on AMX35 was roughly twice as big as the group on placebo. So we had 89 people randomly assigned to AMX35 and 48 people randomly assigned to placebo. And we tested their function and safety over the first 6 months, meaning that they returned to the clinic frequently every 6 to 8 weeks or so for us to measure their function and also to make sure that the drug was safe. Then at the end of the placebo-controlled trial, people who completed the trial had the option to enter what we call an open label extension, which means they had the option to receive AMX35 long-term. And then we checked everyone's survival over a long period of time, over a 3-year period. Just want to start by, in order for us to understand the study results, it's important to understand how we measure physical function in ALS. Some of you may be familiar with this measure because it's commonly used in ALS clinics. The measure is called the ALS Functional Rating Scale Revised, which is also, for short, it's ALS FRSR. And this scale measures independence with different activities using 12 questions over 4 domains. The domains are bulbar, fine motor, gross motor, and respiration. So for example, to assess bulbar function, we ask questions about speech, salivation, swallowing. To assess fine motor function, we ask questions about handwriting, for example. To assess gross motor function, we ask different questions such as ability to walk. And then there's a few questions about respiratory function. So these 12 questions are asked, and then there is a score that's assigned to each of the 12 questions on a scale from 0 to 4, where 4 is better. So the maximum score, which means better function, is 48. Now, what we did was that we followed patients either on AMX35 in red or placebo over a 6-month period. And we saw that treatment with AMX35 significantly slowed down disease progression. There was a significant difference between the two groups, and the numbers are on the slide. So I just want to point out that both groups progressed. So the drug did not stop and did not reverse the disease. However, it slowed down the disease in a significant way. Specifically, people who received AMX35 had the same ALS FRSR total score after 24 weeks of treatment as those in the placebo group did at week 18. So this corresponds to a 6-week increase in retained function. That means that people who received AMX35 were able to retain functions, for example, ability to swallow, walk, etc., longer. In terms of absolute numbers, Again, patients receiving AMX35 had slower progression and a total absolute difference of 2.3 points at the end of the 6-month study. And again, a difference in 2 or 3 points, is a significant difference. Now, again, it didn't stop the disease completely, but this was certainly very, very hopeful news. Also because this was on top of riluzole and edaravone. So these patients were already receiving those drugs. The drug was very well tolerated. We had a small group of people who had a few gastrointestinal events, such as nausea, diarrhea, or stomach pain, especially during the first 3 weeks of treatment. But overall, it was very well tolerated. And actually, people who were on placebo had more serious adverse events, such as respiratory problems, than people on active drug. Now, the newest data, the one that was published most recently, is about survival. So what we did is, we looked at all the 137 participants in the CENTAR trial who entered the trial in 2017 to 2018, and we followed everyone's survival and up to basically this past summer. So people started the trial in 2017, 2018, and then we checked everyone's survival on July 20, 2020. So we compared the people who were originally randomized to AMX35, those 89 individuals that were originally randomized to AMX35, and we compared them to people who were originally randomized to placebo. We had 48 of those. We used public records with a cutoff date of July 20, 2020, which is about 3 years after the initial randomization, to check everyone's vital status. What we saw was that people who were originally randomized to AMX35 lived on average over 6 months longer than the group originally randomized to placebo. So it's a significant survival advantage. And this was independent of baseline use of riluzole or edaravone. So again, this is on top of standard of care. I also want to mention that because of our patient-centered design, as I told you earlier, people on placebo were given the option to enter an open label extension at the completion of the 6 months of placebo-controlled trial. So what this means really is that even the people who were on placebo at some point started AMX35. So we are basically underestimating the survival effect here because most people who were in the trial did get some AMX35, even the ones who originally started on placebo because of this patient-centered design. So essentially the comparison here is really between people who started AMX35 earlier, 6 months earlier, compared to people who started 6 months later. So in summary, in the CENTAR trial, administration of AMX35 resulted in functional benefit with significant slowing of ALS-FRSR and long-term survival benefit with longer survival in the group originally randomized to AMX35. And all of this was on top of standard of care. So I'm going to stop here. And I think we have some time for questions.
If you have any questions, feel free to either unmute yourself or write them in the chat.
I see a couple of questions. Do you want me to read them, or are you, Shannon, are you gonna read them, or?
It looks like they might have gone directly to you because
Just to me. Okay. Yes. Okay, so I got a couple of questions just to me. So one question is, if you did not meet the criteria, is there a way to still be part of the trial? For example, if you have slowly progressing ALS. So if I understand the question correctly, you're asking me that question in relationship to the platform trial. So, and specifically, for example, I think you are referring to the 3 years since onset criterion. So I'll answer that question if I didn't. Yes, okay, exactly. So that was the question. So essentially, there is, as I explained earlier, in the platform trial, there is a time since onset criterion, meaning that people will be included in the trial. One of the criteria is that they need to have disease for less than 3 years. So the reason for that is the following. If people have very slowly progressing ALS and they've had the disease for longer, they don't change as much during a 6-month period. So if people don't change as much because the disease is slowly progressing, well, that's actually a good thing. But in the context of the trial, it means that we are unlikely to see a change over a 6-month period. Now, perhaps we would see a change over, let's say, a 2 or 3 year period, but not over a 6-month period, or not significant. So what that means is that if we were to include people who have very slowly progressing ALS, so people who have had the disease, for example, for longer than 3 years, we would need a much longer and much larger trial to see the treatment effect. So we think the drugs will still work for them, but if we were to include all of these participants in the trial, it would take longer for us to figure out if the drug works or not. We actually quantified that. So we looked at a database called PROACT, which is a database that includes data from people who enrolled in previous trials. And so we noticed that if we were to change the criterion from 3 years to 4 years, we would require a much larger trial, because we would reduce our statistical power. It's a statistical measure, so the power to see an effect statistically by 10%, which means it would take longer for the entire community to find out if the drug works or not. So we basically put a lot of thought and effort and statistical modeling into the design of the trial to try to find the balance between access and speed of development. And so the 3-year mark is what we felt was the best balance to be able to, give access to as many people as possible, while also being able to make the trial fast, so that we can provide results to the entire community. And I do want to mention that if the trial is positive, we expect the drugs to be applicable or effective for everyone, even for people who did not enroll in the trial. Then I see a question about magnetic stimulation, such as transcranial stimulation for ALS, and so one of the attendees wanted to know my thoughts on magnetic stimulation. That's a great question. So that's definitely a field in development. It's sort of a new field for ALS, and I have 2 thoughts specifically about that. So techniques like magnetic stimulation or transcranial, they have different names. Magnetic stimulation, transcranial stimulation, they're sort of part of the same field of medicine, kind of electrophysiology. And that can be very interesting and very powerful as a biomarker or as a measure of disease. In fact, we recently published a trial where we used magnetic stimulation to measure ALS disease progression. This was the trial of retigabine that was led by a colleague of mine at MGH, Dr. Brian Winger, and he recently published the trial. And again, we saw that using transcranial magnetic stimulation can be a very powerful tool to measure different characteristics of the disease. So certainly it can be very helpful in drug development. Now, transcranial stimulation can also be used for other reasons, and this is very new for ALS. So people are trying to understand if it could help, for example, for spasticity or other symptoms of disease, or even to affect like as a treatment, not so much as a diagnostic or a biomarker, but as a treatment. And again, there's a lot of interest in this. I do not know if it's going to work or not, but certainly there are people who are doing research on it. So that's a great question. So I think we'll know more over the next few years once this research is completed. Then I see another question, in terms of, so the other question I have is about Oh, I'm sorry. Just to complete another comment about transcranial magnetic stimulation. So the question was actually about using transcranial electrical stimulation as a treatment. And so the attendee said, I think it helps regrow muscle. Well, there's certainly a lot of interest. I don't know that it's been, again, there's transcranial magnetic stimulation that's for the brain. So that's that basically you put a magnet on the brain. So that one, which is the one that was used for the retigabine trial as a biomarker, and also the one that people are looking at for things like spasticity or treatment of other symptoms. So that's one type of stimulation. Now, in terms of the muscles, actually, there's muscle stimulation that's a little bit different, where you apply the stimulation to the muscles directly. So again, I don't know exactly which one you're referring to. I can tell you that there's research in both. So we don't know if it works. And so again, we still have to see. The last question I see here as a direct message to me was about the center trial and the trial of AMX35. And the question is, as it presented, there was a survival advantage, 6.5 months of survival advantage. And so the question was there quality of life? Well, that's a great question. So we didn't measure that directly in the context of the open label extension study. However, we do know that in the context of the first 6 months of the trial, we certainly had reduced disease progression as measured by the ALS FRSR, which is a scale that measures functional benefits. So that scale tells us that people retained more independence with activities that matter to people, like ability to walk or swallow or talk. So by analogy, I would imagine that that's obviously translatable to quality of life. But again, we didn't directly measure quality of life questionnaires in the long-term extension, but we do know that at least for the first 6 months, certainly we had improvement in function that's generally thought to be correlated with quality of life. I do want to mention that the open label extension trial is still going on. We're about to end it and transition all the people who are still on drug to a separate compassionate use protocol so that they can continue to take the drug long term. Now, as soon as we finish the open label extension that's been going on for over 3 years now, we're going to be able to analyze additional data, and that's going to help answer this question. And also in the open label extension, we did measure the ALS FRSR. We just don't have the final results yet. We're about to really to finish the trial, the long-term kind of follow-up on this ALS FRSR, and then we will be able to share the results over the next few months as soon as we lock the database and we can analyze all the data. And again, I do expect to see positive trends also in terms of functional retention of functional activities or physical functions. So I would imagine that they will correlate with better quality of life. And somebody actually, the last one I have just to me to finish the messages I got to myself is there's a different form of AMX35 be tested for ALS in Italy. Would you consider to be used in combination with the new drugs? So just to clarify, the trial in Italy is a trial of TADCA, which is one of the 2 components of AMX35. So I told you before the AMX35 is a combination of 2 drugs, PB and TERSO, which is also known as TADCA. So I know it's a little bit confusing, but it's basically the same thing. So TERSO and TADCA are 2 names for the same compound. So in Italy, there is one trial that's testing only one of the 2 components of AMX35. So we don't know what the results are. In terms of AMX35, which is one drug that combines the 2, PB and TADCA, we know that it works in combination. You can take it in combination with riluzole and edaravone. So the results will be additive to the other drugs that are already available. And Jen, I see that you turn your video on. So please, if you want to read the questions that were sent to everyone.
Yes, I was going to try to help you out with that.
Okay, thank you.
Thank you so much for your talk. It was wonderful. I think we'll finish up with the questions about AMX35, and then I put a question in about neurome that I thought people would be interested in hearing your thoughts on it. But to kind of summarize a few questions we've got in the group chat here is number one, the FDA approval for AMX35. When might AMX35 be available to the general public? And then the new legislation that just passed the House of Representatives, would that make it available sooner? So kind of like the time frame of what we might be looking at. And I know you might not have all the answers for that, but just any thoughts on that?
Yeah, so yeah, I don't have the final timelines or kind of final answer to all of these questions, because I think it's still an evolving conversation. Obviously, there's going to be discussions between AMELIX and the FDA. We've been part of these discussions as well so far, and I really look forward to the next steps. I think there is a process at the FDA that needs to be followed, and I'm sure that we will be able to provide details, but I'm not trying not to answer. It's really that we don't know yet, because these conversations are ongoing, and so depending on what the FDA says, hopefully we'll know more in early 2021. But again, it depends on the conversations.
If the new legislation, and I'm admittedly blanking on the name of it at the moment, but if it were passed, it has been passed in the House, but it's now going to the Senate next week, and then if it were to be signed by the president, would that make a drug like AMX35 available to people with ALS sooner?
That's a great question. I mean, to be honest, I'm not sure if that specific legislation would help this particular drug, although obviously it's a very valuable piece of legislation, and obviously we all hope that we will pass. But having said that, again, there are already existing pathways for sort of drug approval, and including pathways for sort of priority review, or sort of trying to make sure that things move as fast as possible for a serious illness like ALS. So I'm sure that all of this has been pursued, and again, I don't think it's just a matter of the new legislation. I think it's a matter of kind of continuing the work that has already started with the FDA. But certainly, new legislation that can help ALS in so many ways, aside from a specific drug, I think it's obviously welcome.
I'm going to see if anybody else knows. It doesn't look like there's more questions. Please feel free to put more questions in the chat box, and I will relay those for you. But since no one else has asked it, and I know the community is really interested in this, and I know it's not your study in particular, but it was some compelling data that just was released recently from Brainstorm on their Neurone trial, where they released some of the top line data for that trial. What are your thoughts on that?
Yeah, I was not directly involved, as you mentioned. And my colleagues are, like Dr. Sukovic and others have been working closely on the Neurone trial. So there was some recent, I believe there was some webinars or, some different communications with the community. And again, I don't have, I think this is all work in progress. And in addition to the top line data, it's important to see the detailed data. If you don't know about this, make sure to follow the International ALS Symposium that's going to go on this week. This is the biggest ALS conference in the world, and it's happening this week. And actually, patients can attend, although I think the registrations have closed. But you can check it out, the International ALS Symposium. In addition to sort of the actual conference, they do a lot of work online on social media. There's clips, there's sort of interviews with the presenters. And I know I'm presenting on AMX35 on Wednesday, and Brainstorm is also presenting. Dr. Sukovic and a representative from Brainstorm are also presented on Wednesday, December 9th. So I would expect more press releases, announcements, and just in general, social media material, based on what's presented at the International ALS Symposium.
Also, I think something important to kind of raise about that symposium in particular, we just had a member of the greater Boston area be honored at the Allied Health Symposium as part of the International ALS MND Symposium. John Costello, who is a speech language pathologist like myself, and he directs the augmentative communication program at Boston Children's Hospital, where we refer a lot of you all for communication and assistive technology. He was just honored there as a big contributor to the field. So we're very fortunate to have that expertise that's internationally respected in our backyard. So
absolutely, we love that center and John specifically and all the team there.
What we'll try to do, if anybody's interested in trying to follow along the social media updates related to the International ALS and MND Symposium that's happening, we will try to get that information out to you. Maybe on our social media, Loretta, we could look into that. And if I can get anything, I'll add it in the chat today, so that if you want to start following them on social media and try to find some of those updates and those interviews that Dr. Paganoni mentioned, that we can add those.
Yeah, that's always the biggest event of the year in ALS in terms of the scientific community. It's always the first week of December every year. So,
and I think 3 years ago we held it in Boston.
Yes, that's true.
And had a snowstorm.
Yes, I remember that.
Any more questions for Dr. Paganoni? You definitely have a lot of thank yous in the chat for your presentation today. Thank you so much.
Thank you. Thank you so much.